Hello everybody, welcome to our first remote lab. Today we're going to be discussing RT-PCR and we're going to review electrophoresis, and along the way I'll also talk to you about exactly where you can go on D2L to find all your different resources as well as your assessments that you'll be working on for this week's lab. All right, so let's talk about real-time PCR. I know you guys are familiar with traditional PCR, polymerase chain reaction, and to provide a quick overview of PCR, in our previous labs, we discussed the fact that if you have a DNA sample and you want to amplify it or increase its quantity, what you can do is you can set up a PCR reaction using a thermocycler. And when you do a PCR reaction, it basically amplifies the amount of DNA that you have. And at the end of completing your reaction, you now have not only a lot more DNA, but you're also able to visualize that DNA by doing electrophoresis and looking for specific DNA fragments. The problem with traditional PCR is that you can't really know how much or if you have any DNA in your sample until after the experiment is complete. Whereas if you do real-time PCR or quantitative PCR, we can actually track our amount of DNA as the reaction is occurring. And the reason this can occur is because within our master mix, we're going to be utilizing a fluorescent marker, which can go ahead and bind to our DNA samples, and utilizing a computer connected to a thermocycler, we'll be able to visualize the progress as the samples become amplified each cycle. So part of what our first slide over here says is that when you utilize real-time PCR, you can go ahead and detect and measure your amplified products as the reaction occurs. And this is based mostly to the fact that you're going to be using a SYBR Green marker in addition to your traditional master mix. Now, your traditional master mix, just as a quick review, is where you have your DNA polymerase, your enzyme needed for DNA replication, where you will have your nucleotides, where you will have your primer, and also where you will have your buffers and your coenzyme. Remember that you still have all the material posted on D2L, so if you want to review your PCR, by all means do so. Now, just a little bit about SYBR Green. SYBR Green we have used previously when we were doing electrophoresis. It is an excellent DNA binding dye. It goes ahead and it binds to the double-stranded molecule. And what makes it so fantastic is that when the SYBR Green binds to the molecule, its fluorescent signal is amplified tremendously, making it very easy for us to go ahead and visualize the DNA fragments, even if we are doing things like electrophoresis, or of course if we're hooking it up to the machine and having the machine read our fluorescent signal. The other thing you want to think about is that when you're comparing traditional PCR and real-time PCR, you want to think about what they call the amplification plot. The amplification plot discusses the fact that when we do a PCR reaction, we expect that each cycle will exponentially increase our DNA samples. It will double our DNA samples. So for instance, if I start off with one strand of DNA, after one cycle, now you have two. Do another cycle, now 2 becomes 4. 4 becomes 8, 8 becomes 16, 16 becomes 32, and you just keep going on and on and on. But the truth of the matter is that there will come a time where you switch over from exponential phase to non-exponential phase. And non-exponential indicates that you're still amplifying your DNA, but you are not doubling it each cycle. And this is actually quite common towards the end of traditional PCR, because you might run into issues like your DNA template becomes denatured, maybe your DNA polymerase enzyme is no longer functioning, maybe you don't have enough nucleotides to amplify the DNA, or maybe it could be something that your primer are malfunctioning and it can't find a distinguished fragment that you're looking for. Now, the non-exponential phase can't be detected in traditional PCR. In fact, you won't be able to quantify any of your DNA product in traditional PCR until after you've completed the experiment. On the other hand, when we're doing our real-time or qPCR, what we see happening is because we have that fluorescent signal, we'll be able to track the amplification plot, and we'll be able to narrow down when it switches from exponential over to non-exponential replication. In addition to that switch, we'll also be able to calculate what we call the CT value, which is the threshold cycle. Your CT value indicates how many cycles of PCR it took before you had enough DNA for the fluorescent signal to be detected by the computer. And usually what we see happening is if your initial sample had a high amount of DNA, you will have a low CT value. It will only take a few cycles for you to have enough amplified DNA to be detected by the computer. On the other hand, if your samples had low amount of DNA, what we see happening is that you would have a high CT value. And that would indicate that you had to do a lot of replication cycles before you had enough DNA in your product for the fluorescent signal to be picked up by the computer. Another item that we can have exposure to when we do our real-time PCR is what we call our melt curve analysis. Our melt curve analysis allows us to distinguish between our products and any non-specific products, such as maybe contaminants or abnormalities or different size fragments that we were not interested in. When you complete a melt curve analysis, the computer will keep track of the behavior of your product as it cycles through different temperature points, and it will represent it in a graphical form. This is what you see in graph number A. And as we're seeing along, we see that our samples are following very similar pathways as the peak goes up and it comes down. If there were any lines forming that were irregular and not following the flow, that would be a really good indicator that you might have some irregularities, some abnormalities, or maybe just some contamination within your sample. Another thing I wanted to mention to you, and this part you didn't complete in lab, is the fact that you have to, in essence, teach the computer to recognize your DNA sample and the particular primer that you're working with. So in order to do that, we had to go ahead and complete a serial dilution. This was done before you guys actually came to lab, and that way it kind of saved us a little bit of time. But when you do your serial dilution, you basically have a DNA template that is mixed together with your primer of interest in various concentrations. This allows the thermocycler and the fluorescent signal, in essence, to get used to the amplification curve of that particular primer. So you can go ahead and you can set up your dilutions and you can make your melt curve analysis. And as we're analyzing our results, it will also allow us to learn more about our particular primer by coming up with a log behavior. And this, of course, will lead us to create a standard curve. The standard curve, which will be represented in a linear regression line, will allow us to follow the DNA as it's being amplified and being able to recognize the particular DNA fragments that we would like to increase. Now, please keep in mind when we were doing our GMO experiment, you had your green primer, your PMM, and you had your red primer, your GMM. The PMM is the one that was our quality control to make sure that we had plant DNA in our samples, and the GMM was for our GMO detection, utilizing promoters and terminators that were found in about 80% of the GMOs currently on the market. So here's just a quick reminder of what we did in our last lab. You had to extract DNA from the oats. That was our negative GMO control. You had your positive DNA already extracted, and then you had your food samples. And each of us went ahead and completed our samples by mixing them with the PMM and the GMM. And as you can see from the template on the right-hand side, it just reminds you that we grouped together the PMM, those were the first four, with our oats in A. B and C were our food samples, and D was our positive control. And then we followed the same pattern, oats, food, food, and positive. We followed that same pattern for E through H, and that was with our GMM or our GMO primers. Now, what we were supposed to do in our actual face-to-face lab last week was to take our results from our qPCR and run electrophoresis on it. And we were to follow the same protocol as we previously did. Once we had our products back, our PCR tubes back, we were going to go ahead and add orange G loading dye to it. Remember, orange G loading dye will sink your items to the bottom of the well because it makes it heavier. It increases the density. And then we were also going to add a little bit more SYBR Green to our samples to make sure that we would be able to follow the fluorescence in the electrophoresis gel itself. On the side over here on your left-hand side in the text, you can see that I went ahead and I reminded you that your red primers had fragment sizes of 203 and 225. Your green primer for your photosystem gene, that one had a fragment size of about 455 base pairs. You also have the use of your PCR molecular weight ruler that we can utilize to standardize our different sizes and their location, making it easier for us to identify our 200 and 400 base pairs. Now, I know we weren't able to complete our second electrophoresis, but I definitely wanted to go ahead and show you this picture again. This was one of the gels that we had the last time when we did our traditional PCR, and it just basically shows you the fragment sizes fluorescing due to the SYBR Green, and our job, of course, was to line up the fragment sizes according to their sizes, utilizing the PCR marker, and then being able to indicate if the fragment was there for our PMM, indicating there was plant DNA in our sample, and if the fragment was there for our GMM, that meant that the DNA had a GMO component to it. So the question now is, since we're switching over to an online module, how exactly are we going to be doing our electrophoresis? Well, we won't be able to do electrophoresis on our actual samples because we have no access to our lab. But instead, what we're going to do is I've loaded a bunch of different activities for you to complete and to get some more practice on electrophoresis. So the first thing you need to do is go ahead and log into D2L and select your 3063 shell and look on the side menu for the module titled Online Module, Lab number 8 through 9, RT-PCR and Electrophoresis. And when you open up that module, you're going to notice you have a wide array of different activities that are on display. The top one will show you your PowerPoints and your protocols, so please make sure to review through that, get some more background information, take a look at the PowerPoints again if you'd like. And then I've posted a bunch of different videos for you. The first video, which I entitled Video Required, Bio-Red Presentation on GMO Investigator, Real-Time PCR Starter Kit. I would love for you to watch that video. It is about an hour long. It is done by Bio-Red, which is the company that actually does the GMO kits that we were utilizing. In the video, they will discuss the purpose of RT-PCR, and they will also discuss how you can analyze all the different results. Please make sure that you watch this video because I will be taking some questions, or I will be taking some of the material and making questions for our quiz that we'll be having next week. Underneath the required video, I've posted some additional videos that I've come across on YouTube that I found helpful. Please keep in mind those two videos, and I've indicated it for you in the title. They do not have any audio, but they're really good on animation and exploration side in text. There's also an overview for PCR, and then as we make our way towards the bottom, you're going to notice that you have an activity. I want you to go ahead and follow the link posted to do the virtual electrophoresis activity. And once you have completed the activity, there is a worksheet that goes with it. So go ahead and complete that worksheet that can be located in our lab number 9, RT-PCR and Electrophoresis assignment folder, which is all the way at the bottom of this illustration. So go ahead and get the worksheet, fill it out, and then please submit it. And you have until Wednesday, March 25th at 11:59 p.m. to do so. And I'll get that graded for you. Now, guys, as a final note, I know this format is new for many of us, so I really do encourage you to keep reaching out. And if you have any questions, comments, and concerns, I am literally an email away. And I will also open up a discussion session that you can go on and you can post all your questions and your comments. I would love for our student engagement to continue on, so please don't be a stranger. And if you're not sure about anything, please email me and I'll get back to you as soon as possible. All right, have fun.